Stanis, and could you
Hi,
please
how are you?
introduce yourself? Yeah, hi.
Okay, so I'm glad to hear and see everyone who will listen to my talk online and also offline. So my name is Stanislovas Jankauskas. I'm originally from Russia. There I received my PhD. Then I did my several postdocs in Germany and United States. And so today I will tell you about the experiment that is funded by the Open Longevity and will be performed by me on the basis of Albert Einstein College of Medicine in New York City, where I work now. And it will be the combination of mitophagy agonists, Senolytics, and anti-glycation therapy for lifespan extension. So, okay, so let's start with the question. So why should we fight aging? So the aging is one of the most serious long-standing problems of the humankind. So on the left graph, on the left figure, you can see how the share of young people from like kids and teenagers is shrinking from 44% in 1950s to 27% as it is expected to be in 2050. And so their share of the people older than 65 is increasing like more than 3 times from 5% 70 years ago to 16%. This means that every 8 person in the world will be soon quite old. And you can think that there is no big changes if you look on these, green part of the square of the circle. It changes from 51% to 57%, but the share of people who are closer to 64 is much bigger in this green area. And on the right, you can see these nice animated population pyramid from Denmark data. So you see that in 1950s, so the amount of the old people was very little, and it was much smaller than the amount of the young people. And now you see that the situation totally changed. So now we have the amount of the elderly much bigger than amount of the young people. So you can agree that the world population is aging, but so you can still ask, why it's bad. So it's another way to ask, why should we fight aging? And there is at least 4 obvious reasons. So one reason is that aging decreases the quality of life. So here you can see the data from the US. So it's a self-reported life satisfaction, and we see that it's gradually decreasing with the age. So the second reason is that aging, the ultimate reason for our death. So on this graph, you can see the probability of death during the next year of your life. And we see that it's gradually increasing from like it's 1/10 of the percent. So when you are 20 years old to more than 50% when you become extremely old. Of course, during your life, you can die from infectious disease, you can die from some accidents, you can be a victim of the crime. But if you will be lucky and you will, escape all the dangerous situations, so ultimately you still will die when you will become old. So aging is associated with higher risk of diseases and with their poor prognosis. So on the top figure, you can see that prevalence of many diseases is increasing with age. And maybe what is even more important, that all people become very fragile, and many diseases that are not dangerous for younger people became lethal to the old people. So the most famous example is, I guess, is a COVID. but I will show you another data. So it's the data from the Germany. It shows the mortality for myocardial infarction for men, for women, so during the different years. And so you see when myocardial infarction occurs in the man aged from 25 to 54, the mortality is quite low, and then it's increasing like 5 times when you become 10 years older, and then additional 3 times when you become additional 10 years older, and when you become extremely old, this mortality becomes very high. So actually, so the difference in mortality between people who are more than 75 years old is hundreds of times higher comparing to the people who are under 54 years. And so you can see, so during the last years, we have, some progress in, managing the extremely ill people. So we see that both men and women, the mortality is increasing, but it's difficult to imagine that, with this trend, we can lower the mortality of old people to the level of young people. So we need to cure something that underlies these huge augmentation in the probability of death. So the fourth reason could sound, a little bit pragmatic and inhumane. So aging is a problem for economy, and especially, quite inhumane. So this figure, could look quite inhumane, but it's a real problem. So the old people, still maintain their consumption, but they do not earn money. And if you look like how societies cope with this problem, you can see that, for example, some countries like Asia, so they get lots of money from their kids supporting their parents. But how to say, this situation wouldn't long forever, because as we see, so the share of young people is decreasing. So United States are still benefiting from their very good retirement system and their unbelievable economic growth during the 1960s and 1980s. For example, so in the European countries, you see that the majority of the money that is used to prevent, to be honest, all people from starving is going from the government. And actually, if we could fight the aging, we can make people, we can give people much more energy. They, could be still working. They could be still, good professionals. They wouldn't be so tired, so fragile. And so it's a really good thing for economy. So if the aging is so big problem, so why we still don't have the answer to this problem, answer to this challenge? And the reason is that the aging is a multifactorial condition. So during the 20th century, there was many attempts to make one single theory of aging to explain everything that happens with organ interengagement with one, single process. For example, accumulation of somatic mutations, accumulation of mitochondrial damage due to the oxidative stress. But now in the 20th century, we realize that it doesn't work. So that there is many things that contribute to the aging. So first of all, we know that there is no single gene, or maybe 2 genes or 3 genes. There is no, just several genes that control the aging, and we know this because we don't have any examples in our written history of people who lived very long, who lived for hundreds of years, or people who did not demonstrate any signs of aging. And the amount of the people that live so far on the earth is estimated to be 120 billions, and even if we subtract the people who lived on the earth during the times when we have no written history and we'll take only the people who lived during the well-documented time, we will have 4 billion people. And in every generations, there mutations, sporadic mutations happens. We all have lots of mutations. And actually, so it means that, nature had around 40 billion, attempts to find, the gene that control aging. So the nature was, probing dice for 40 billion times. And the fact that so we never saw, a man or woman without aging, saying that one single mutation or maybe even 2 mutations, so they do not change this phenotype. So we also know that there is no single therapy or diet or lifestyle that creates radical extension of life. We know this because when we look for the people who live then more than 100 years, so they came from different regions, so they have, different genetic background, they have different diets, they have different lifestyle. And these diversity says us that there is no one, single life hack that can fight aging. And we see the same in our experiments on mice. So far we have maybe like several dozens of drugs that are able to produce the lifespan of mice. But none of them can do this more than 40%. No one, none of the drug can give more than 40% for the lifespan of mice. And this also, matches so our conclusions from what we see from the people. So affecting, only one thing, so wouldn't give us radical life extension. So you may say that all this is very pessimistic and it looks like that we cannot fight aging. But on the other hand, if we look on another data that we have from the real world, we can say that we can be optimistic. If we look on these data from the paper who looked at the ecology and evolution of the aging, so you can see the graph that shows the relationship between the body mass and the longevity. And each dot is a single species, so we have, lots of species, and it's one of the general rules that is well known for people who study aging, that there is a strong correlation between body mass and the longevity. So the bigger animal you have, the longer it lives. But despite this rule, if we, for example, take some, body weight, like here, like 10 grams, and we will see so what the diversity of the longevity we have. So some species with their body mass around 10 grams can live around, 2 years, and some species can live more than 30 years. And so these 10 times difference, so within the same body mass, tell us that the aging is not something inevitable. It's not only, some inevitable, physics processes or chemistry processes. It's something that organism can regulate, something that organism can adjust. And one, very interesting finding of this paper. So they found that so one of their very strong predictors about the longevity for the species is the presence of wings. So the red is non-volant birds and mammals, and blue is volant birds and mammals. And you see that mammals without wings, their aging is faster, so their longevity is less according to the same body mass. So people do not have wings, but maybe we can invent some wings. So why shouldn't we fly? So another set of data that also makes us very optimistic came from the assessment of longevity So here in green, and rate of aging in yellow. For reptiles and amphibians, it's a recent paper from the Science. And the most interesting thing is the rate of longevity. So it's here represented as a beta. So yeah, I'm very bad at math, so I didn't put this equation, but people who are good in mathematics, they know that so these curve that show us the probability of death for the next year. It could be very good described with a so-called complex equation. And there is in this equation, there is this factor beta that actually so defines the angle of the slope and actually so the rate of the aging. So how, how fast the probability of death will be increasing with the age. So in the majority of the countries, for the rate of aging for the people would be around 1.6. And for example, so if the beta is increasing, so it means that the aging is faster. When the beta is decreasing, so this means that aging is slower. And it's very so striking thing that if you look at the reptiles and amphibians, you can notice quite a lot of species for which the beta is close to 0. So this means that there is a lot of species, they have very slow aging that could, or maybe even neglectably slow aging. So they have, these, so this curve will be almost flat. So there is no increase in probability of death at age. And some species, they even have negative values of beta. This means that the probability of death with the age for these species is even decreasing. And so this says that biological organisms are not only capable of adjusting the aging speed, aging rate, they can even stop the aging. Okay, so going back to the problem with the aging is that it's a multifactorial condition. So I use this picture from one review that telling about the DNA damage, role of DNA damage in the aging, and so it's it's only one aspect of the aging. But so you can see, so in this picture, so it's very nicely represents the problem, is that the processes that involve aging, they're interconnected. So DNA damage induced metabolic dysregulation, cellular senescence, mitochondrial dysfunction, it decreases autophagy, and metabolic dysregulation can also cause cellular senescence, also decrease autophagy, also cause mitochondrial dysfunction. And the problem that if you will be targeting only one component, so you wouldn't be able to fix the problem because, for example, you will touch the mitochondrial dysfunction. But autophagy, decreased autophagy will be contributing to metabolic deregulation, and metabolic deregulation will be contributing to cellular senescence, and cellular senescence will be playing against you in when you are just trying to decrease the mitochondrial dysfunction. And it's the reason why we cannot fight the aging using only only one genetic therapy or only one, medication. So we need to simultaneously act on different things in the aging phenotype. So we actually need the combination therapy for the aging. And actually, so it is the thing that is already widely used in the medicine. So here you can see the two pictures from two reviews, both published in the Cell. I guess both of them published in the same year, 2022. And you see that they are pretty similar. So it's the hallmarks of aging and it's the hallmarks of cancer. And you can see that, the picture is the same. So here's, the circle with the different hallmarks, the same here. And so in the middle of the circle, the process, because all these factors are contributing to the process. And could you say what's the main difference between these two pictures? The main difference is that the picture for the hallmarks of cancer have additional circle. The circle with established therapeutical approaches. They target each of the hallmarks of cancer, and we don't have this for the hallmarks of aging. And so this thing actually gave us enormous success in fighting the cancer. So the idea that we should simultaneously act on different parts of the cancer mechanism, gives opportunity to make a progress in this disease. So here you can see the number of deaths in the world for men and for women from oncological disease from 1975 till 2016. And you see so how this number of cancer deaths was increasing during the from 1975 to 1990s, and it's the blue line, so it's the real mortality. And the red is the extrapolation. So if you look at these 15 years and make extrapolation, you will see so this increase. But actually, so from the 1990s, these increase in cancer death, become much slower. Because we established lots of combination therapies for treating cancer. So in this table, I gave examples of some, very famous combination therapies. Two of them are for the cancer, and so the idea that you use together simultaneously the drugs with different mechanisms, for example, alkylating agents, anthracyclines, mitotic inhibitors, corticosteroids. But also we use, for example, combination therapy for HIV. And so this is an example of one of the combination that includes two different nucleoside reverse transcriptase inhibitors, protease inhibitor, pharmacokinetic enhancer. And also, for example, so another disease that is treated by the combination therapy is hypertension and atherosclerosis. And atherosclerosis. For example, so we use together calcium channel blockers, angiotensin receptors antagonists, diuretics, and statins. So that's why we want to perform an experiment in which we will use the combination of drug of different medications that will target different hallmarks of aging. So in the beginning, I will describe the design of the experiment. So we will have two groups consisting of 30 male mice, C57 Black 6J. So they would be at the age of approximately 1 year. They would either stay on the normal standard lab show, or they will receive their drug combination. And we will look at the survival of the animals. So the endpoint will be mice demise. And we will perform non-invasive measurements before the initiation of the diet and when the animals will reach 50% lethality in both groups. So we hope that it will be actually chronologically 2 different time points because we hope that our combination will increase the survival. And we want to not to do the non-invasive measurements in the same chronological time point, because there is a reasonable skepticism about looking at the aging only, looking on the assessing the effect of anti-aging therapy only by the survival curve. So these arguments points that if you will treat the disease that is most likely causing the death in this animal, you can see the increase in survival without affecting the aging. And it's very easy to understand if you think about the modern medicine, when we are able to treat many diseases in all people. And so treating this disease, we prolong their lifespan, but these people do not become younger. So of course, it's good. So to save lives of these people, but much better would be to make them younger. And so that's why we want to perform these measurements not in the same chronological time, but when they will reach the 50% survival. And if there will be no difference in these parameters when they will reach 50% survival, it will be saying that most likely we did not achieve our goal. Of course, the great limitation of our study is that we have only 2 groups, because we have quite vast combination. And in case of success or unsuccessful combination, it would be not possible to say. So if the combination is redundant, if there is, we can, get rid of some drug and you will have the same effect. But so the problem is that such experiments are very expensive. And so our budget is very limited, and now we can afford only 2 groups, but so we look forward for we would be grateful for any who can support our experiment in order to add additional groups, or to get better understanding of the effects of the combination, or even maybe to try another combination. So let's go to the description of the drugs that we want to use and why we want to use them. So the first one is enalapril. So it's is the safe medication that is used in people for, I guess, more than 20 years. It's used for treatment of hypertension, but then it was also found that it's also good in treating heart failure, in increasing survival of the patients with chronic kidney disease. And experiments in mice and in rats shows that enalapril can prolong the lifespan of animals. So why it's happening on the mechanistical level. So here you can see the signaling of metabolism and signaling of angiotensin. And enalapril actually it's ACE. It's inhibitor of angiotensin converting enzyme ACE, or ACE1, because there is also ACE2. And so actually angiotensin 1 is converted by ACE into angiotensin 2, and angiotensin 2 interacting with its main receptor H1R causes vasoconstriction, hypertrophy of cardiomyocytes, it triggers inflammation, oxidative stress, and fibrosis. It also acts on the AT2R, which has actually the opposite effects, but so usually the AT1 receptors are more abundant than AT2R. And also there is alternative pathway when angiotensin 1 is cleaved by ACE2 into angiotensin 1,9 and angiotensin 1,7. And so these compounds are acting, they're not acting on angiotensin 1 receptor, which is deleterious. Of course, it's deleterious in terms of chronic activation because, in the terms of acute activation, of course, we need angiotensin because it allows us to mobilize during the stress conditions to constrict the vessels that supply the blood to the organs that are, not so important during, the survival in external conditions. But the chronic inflammation is bad. So let's name these receptors deleterious. And so the products of ACE2, they're not interacting with AT1R. They're interacting with AT2R, with MAS, and with another receptor, which of them has their opposite effect to angiotensin 1 receptor. And actually, so ACE2 become, very famous recently because it's one of the receptors for SARS-CoV-2, which is causing COVID. And actually, so one of the mechanisms of the COVID is the inactivation of ACE2 by the virus, and actually, so deleting all these good byproducts of angiotensin metabolism. And so using enalapril, we will decrease the amount of angiotensin 2 on the one hand, but on the other hand, there will be much more angiotensin 1 available for ACE2, and we will be stimulating the good receptors of angiotensin. So here on this picture, you can see it. So how angiotensin receptor 1 is producing reactive oxygen species, and it has lots of deleterious effects. It damages mitochondria, it damages DNA, proteins, lipids, and contributes to the aging. And also the interesting thing that angiotensin is known to be very strong agonist of cellular senescence. So, for example, so I just give you from the papers, but actually there are many papers showing this. So the P16 is a marker of cell senescence, and you can see how much P16 we have in the artery when the animal is injected with angiotensin 2. And here you can see the effect of angiotensin cell culture, and with blue you can see the staining for X beta gal, which is another marker of senescent cells. So using enalapril, we can definitely act on mitochondrial dysfunction, preventing oxidative stress, and we can prevent cellular senescence. Okay, let's go to the second drug, everolimus. So everolimus is actually the analog of rapamycin. It actually was developed later than rapamycin to get a drug that would be more specific in inhibiting mTOR. Actually, so everolimus is also used, it's a medication that's used in people, and it's mostly used for the treatment of distant oncologic disease. Okay. So rapamycin is actually so one of the most prominent drugs in the aging research because it has very strong effect on physical longevity in many species, and actually so it acts via these proteins called mTOR, and actually so in the beginning so the pro-longevity effect of rapamycin was discovered, and only then it was discovered that this effect is working through this protein, and this protein was named after rapamycin. It's actually called mechanistic target of rapamycin. So rapamycin, mTOR makes two different complexes, mTOR C1 and mTOR C2, mTOR complex 1, complex 2, and so the complex that is most associated with aging is C1, and rapamycin acts on both of them, and everolimus only on mTOR C1. So there is mTOR really lying, in the middle of aging processes. You can see it has, very 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 complicated the net of the signaling pathway net with different sensors, different effectors, and mTOR controls lots of processes that that are connected with aging. So it's controlled energy homeostasis, cellular senescence, stiffness of the cell, proteostasis, and so on. And so as I told, so the problem with rapamycin, why it's not used already as a treatment for the aging, because it has lots of side effects. So one of the most common side effects is the glucose intolerance that is caused by rapamycin. So when you check glucose, you see how fast the level is increasing, and when you the open circles is animals with just weekly treatment. The black squares is the rapamycin. So you see that the glucose, when you check the same amount of the glucose, it's clearance from the blood is retarded, and the glucose reach much higher level. And with everolimus, this negative effect is much smaller, and also everolimus is not so strong immunosuppressor like rapamycin. And one of the most stunning things about rapamycin and everolimus that the effect could be extremely long. So one of the main effects of the rapamycin, and which is related to the aging, is activation of autophagy. So you can see the marker of autophagy here by the colocalization of green and red. So this yellow or orange, and you see that in the control mice that receive no treatment of the age of 1 year. So like you see, this small amount of autophagy marker. And when you treat the animals for 9 months with rapamycin, and only for 3 months for rapamycin, then you make 6 months pause. So you have the same effect on the activation of autophagy. You can withdraw rapamycin for 6 months, and its effect is still present. And it was also found in the study when the lifespan effect of rapamycin was studied. So the green line is the survival of control animals. Dark red is survival of animals with that were constantly receiving rapamycin, and blue is the animals that receive rapamycin for 3 months, and then it was discarded. And you see that longevity effect is the same. So everolimus would act on different hallmarks of aging. It will counteract disabled microautophagy. It will normalize the proteostasis. It will be fighting with regulated nutrient sensing. It will be decreasing cellular senescence. It will be counteracting stem cell exhaustion. Okay, so the third drug in our combination is sulforaphane. So why we choose this drug? Because this drug, sulforaphane, is an inhibitor of protein called KIAP1. And why it's important? Because KIAP1 actually making to degrade another protein, NRF2. And when sulforaphane is binding with the KIAP is not able to degrade NRF2, and then NRF2 goes to the nucleus and binds to the DNA because it's a transcription factor, and it controls the huge amount of genes which are very beneficial for the cell. It controls the it actually triggers the expression of many antioxidant genes that are related to thyroidoxin metabolism, glucagon metabolism. It also triggers the expression of the genes that are involved in iron metabolism, and when the genes are activated, the amount of the free iron is decreased, and free iron exacerbates the consequences of oxidative stress to very high degrees, and even iron in combination with the oxidative stress makes the specific, induce the specific type of cell death called ferroptosis. And beside these good effects, another interesting feature of NRF2 and also the sulforaphane, because sulforaphane increases the amount of NRF2, is that NRF2 competes with NF-kappa beta. So NF-kappa beta consists of several subunits like P60, P65, P50, and they actually compete for the same consequence of DNA. When you have lots of NF-kappa beta, so NRF2 is not able to bind to DNA, and its genes are not. And otherwise, when you have lots of NRF and RF2, NF-kappa beta is not binding to DNA, and they also, have some kind of they were a proxy, for example. So NF-kappa beta is able to activate histone deacetylase free, and it deacetylates the DNA around NRF2 and prevents the expression of the genes. And NRF2, for example, increases the expression of gamma oxygenase 1, which inhibits NF-kappa beta. So it's really big war between them. And NF-kappa beta is the master regulator of pro-inflammatory genes, and it's in terms of aging is very important because it induces the expression of genes that actually contributes their so-called senescent associated secretory phenotype. So it's the number of when cells encounter some damaging processes, they able to get into so-called senescent state. During this state, they're not able to proliferate anymore. And they also start to secrete a specific set of the proteins that also triggers the senescence in bystander cells. And it's one of the important mechanisms of the aging. So cell senescence is somehow contagious. So once you have, little amount of the senescent cells and the immune system is not able to get rid of them, is not able to kill them, then you have more and more cells around this initial senescent core to become senescent. And one of the way to stop these contagious spread of senescence for the tissue is to prevent the secretion of these proteins, and this is controlled by NF-kappa beta. So that's why sulforaphane will decrease the chronic inflammation, will prevent cellular senescence, and also will contribute to the protection of mitochondria. Okay, so the fourth compound in our combination is NAD. It's a nicotine adenine dinucleotide. It's contained in literally in every cell, because it's one of the central coenzymes to metabolism. So many processes are helped happening with the help of NAD. And there is, huge amount of the papers showing that decrease in NAD concentration is one of the features of the aging. So in many tissues, like I think maybe in all tissues, the NAD is decreasing with the age, decreasing in different species. So there is also lots of data, lots of papers describing that supplementation of NAD has lots of beneficial effects. So supplementation of NAD prevents the so-called polarization of macrophages. So macrophages could be, so in the two, they have actually, how to say, two states, M1 and M2. One is very inflammatory, so it's like active macrophage that release lots of reactive oxygen species, nitrogen species that kill all the cells around them. And another phenotype is like much more, present macrophages. And supplementation of the NAD, so reduce this amount of pro-inflammatory macrophages, which is good in terms of the aging, because in aging we have accumulation of different inflammatory stimuli that contribute to the persistent, not ending chronic inflammation. So NAD supplementation was found to cure the neurodegeneration. So NAD so is actually the substrate for the cytoplasmic deacetylases that is called sirtuins. And sirtuins, by deacetylating a number of proteins, are stimulating their autophagy. That is a very important problem, because it is the key processes that allows cell to make a quality control of the proteins, of the organelles. So if the protein are not working good, if the organelles are not working good, so cell utilizes autophagy to get rid of the cells. And by adding NAD and stimulated sirtuins, we can enhance these cell clearance. NAD also counteracts the circulation of epigenetics that happen in aging. And also NAD is important for the proteins that participate in the repair of DNA breaks. So NAD would counteract such hallmarks as genomic instability, chronic inflammation, epigenetic alterations, disabled macroautophagy. So the fifth compound love combination is the nobility. It's a natural compound, and there is lots of good data, both so in different species, like in nematodes, C. elegans, and also in mice, showing that nobility can prolong lifespan both of C. elegans and mice. And it was showing that it has that it's acting on very important pathways for the aging. So in C. elegans, it was found out that it's activating so the SIRT. So it's so this gene in C. elegans is named like this, SIRT2, but it's the same sirtuin deacetylase. And it's also increasing the genes that are connected with the oxidative stress. And in mice, it was showing that nobility contracts the NF-kappa beta by acting on the factors on the proteins that controls the circadian rhythm. And it's there is a very good paper showing that so the regulation of the sleep, when people cannot sleep, or due to the lifestyle, or due to their work regime, their work hours, they have that dysregulation of circadian rhythm. This contributes to the development of the chronic inflammation. And so nobility in mice was shown to normalize the circadian rhythm and thus stop the chronic inflammation caused by dysregulation of circadian rhythm. So, nobility will counteract the chronic inflammation. It will counteract cellular senescence, the mitochondrial dysfunction, and disable mitophagy. So, the last three compounds, they could be grouped into one mechanism. So it's so all these feedbacks they are could be named as anti-glycation agents. So they are very different. So the pyridoxamine is the form of vitamin B6, tannic acid is a natural compound, spermidine is the product of amino acid degradation in the organism. And so we need these feedbacks because there is a such deleterious process in the organism as the accumulation of end products of advanced glycation. So it's a set of consequent chemical reactions that actually results in adding some specific groups to the proteins, and these additions they disrupt the normal protein structure, like this. And so maybe the most important problem is that ages they advanced glycation products, they bind to the proteins that comprise the extracellular matrix. The extracellular matrix is a very important thing. It's like some kind of the skeleton, and it's much more than the skeleton. It's actually so the milieu for the cells, so the cells understand so that they are located in the right place by interacting with the matrix. So if the cells are not interacting with the matrix, so usually they go into the apoptosis. Actually, so the cancer cells, so it's one of the features of the cancer cells, they turn off these induction of the apoptosis when they are not connected with the matrix, and this allows cancer cells to give metastasis into different organs. They can travel. So normal cells, they are not able to travel. Also, the cells, when they differentiate, they also understand. So they need to differentiate, and to what type of the cell differentiate, they understand this also by interacting with extracellular matrix. Extracellular matrix also makes the proper physical parameters of the tissue. So it makes the tissue more rigor or less rigor. It allows tissue, to be stretchable or, not stretchable at all. And age is actually interacting with the proteins of extracellular matrix. They compromise all these functions. So the extracellular matrix become more rigor. So the cells start to receive their wrong misleading signaling. And so we need these feedbacks because they are interacting with the different chemical reactions that are producing ages. And so we believe that actually, so the age-mediated extracellular matrix alteration is very important hallmark, but so in the most conventional paper, so about the hormones of aging, so they did not put the alterations of the extracellular matrix as a hallmark, but we, according to our data, we believe that it's very important to also contradict these hallmarks. So in general, so we have a combination in which we are targeting the majority of the hallmarks of the aging, and we also are targeting the different. So we also touched some hallmarks. So with the several drugs using different mechanisms, and these. This is our idea how we can develop a therapy that could radically extend the life of the mice, and then we hope the human. It will be the end of the talk. So I would like to present our team. So for this experiment, so for this project, we have a scientific committee consisting of me, of Roman Litvinov, who is also the head of the research project Boston Matrix of the open longevity. So he's the specialist in anti-glycation therapy. I don't know if he's listening this work, but if you will have, some comprehensive questions about how anti-glycation therapy will work, so I will ask Roman, and he will, I will redirect questions to him. So also we have Alexey Rashevsky in our team, who is he has extremely broad expertise in different aging mechanisms. So the experiment will be performed by me on the basis of Einstein College of Medicine. So here the head of the open longevity, and I will be happy to answer any questions from you.
Stanis, thank you so much. That was great. I actually have a question. Could you please go back to the slide where you show the list of all the compounds and how they affect all the hallmarks? Yeah. I can see that there are several hallmarks that we are not affecting at all, or maybe not affecting directly. Do you think that this gives us like some major risks, like we're not affecting telomere attrition, and also what's alters intercellular communication? That will be mildly affected, but dysbiosis, like things like that. What do you think? Do we need to affect them all to see the result?
I think that ideally we should affect them all, but we have several limitations. The major limitation is the budget. That for example, it will be nice, for example, to target the telomere attrition, but at the point we don't have any pharmacological tools to do this. So for this we need to use genetic therapy, and for a pity with our budget we cannot afford using this therapy. Another limitation is that the aging affects very different aspects of our organism. And the problem is that we also need to broaden our scientific community, because actually, regarding the dysbiosis or altered intercellular communication, I think that we don't touch this because we don't have expertise in this. So if we can get on the board people who have good expertise in this, so maybe they can suggest plus some tools. Of course, how to say, not so expensive. So I'm talking about the money, but it's the real. So it's maybe one of the greatest limitation in the Jank studies, because even if you're doing some academic research, it's already very expensive, and that's why, so it's much more expensive than doing cancer research. And so that's why I'm pointing so many times of the limitation of our budget. But yeah, so the reason why we do not touch it,
Yeah, we need
this,
experts
yeah,
in this
yeah, this,
area. Mhm.
yeah, this, because yeah, we don't have expertise on these, and so we need experts. But in general, so I think that maybe targeting maybe 2/3 or even the half. So if we will find the way to touch it, maybe the half of the hallmarks at a very good level, I think we still should have the good effect, more than 40% of increase of the lifespan.
Mhm. Great, thank you. And could you please go back to the one of the first slides? Well, it's somewhere in the middle. The whole design, like how much time does everything takes?
Yeah. Okay. So.
Yeah, here. So the graph, like sort of in the middle and the bottom, the two lines, the blue and the gray one. This is the therapy and the control.
Yeah, so we will have two groups. And here, so it's how to say, the optimistic view of how we see the results. So for the blue line is, quite a realistic representation how it happens. Usually, so the half of the animals, they die at the age of 800 days. And so the maximum lifespan for the mice is about 1,200 days. But how the curve lie for the treatment group, we don't know. So maybe it will be very close, maybe it will be not so close. But we will start our experiment when the mice will be around 1 year old, around 400 days. And we will wait until they all die, and so it will at least take 2 years. But if our combination will be very good, then maybe we should wait for 3 or maybe even 4 years. Yeah.
And we will make this non-invasive measurements on like at the very beginning, obviously, and then like in the middle of their like survival rate dropping, right? Somewhere like at year 2 point.
Yeah, so the idea is that because we are limited in the budget, so we don't want to decrease the number of the animals. So we want to use all 30 animals to see the survival, because if we'll be sacrificing at some points, the animals will have decreased, will decrease the number, and maybe we will lose we will lose the ability to prove statistically that we have effect. And also why we will be making non-invasive measurements only 2 times, because from my experience working with the old mice, they're very fragile, and I don't want to put additional stress on them.
So
yeah, but yeah, of course we will be collecting the corpses of animals, and if it will be possible, we will we'll try to analyze to get the information that we can get from the dead animals for like mRNA transcripts, or like analysis of the proteins using Western blot. Maybe it wouldn't be so good, but for example, for the histology, for example, yeah, we can. I think we would be able to distinguish between the postmortem changes and the changes that were induced by the agent.
Okay, so it says non-invasive measurements, but in fact, how them is invasive. It's just not harsh, like urine, like glucose tolerance tests, or some blood collection. Yeah, okay, right. And most of it is like behavioral tests. I'm guessing there's a question in the chat. March is asking, will you pay attention to behavioral patterns of animals during experiments, in particular their physical activity, signs of apathy, dementia, and so on? You, by the way, can ask your question if you want, if you need some clarification. It's not only me who is allowed to speak, it's fine. Please raise your hands. But yeah, I'm guessing the answer is yes. Behavioral tests are planned to be.
Yes, we will try to assess these things. Okay, so actually, so I don't have expertise in neuroscience, so I cannot say at this point. So if old mice really develop, you know, dementia and apathy. So as far as I know, so there is no lots of neurodegeneration with the Asian mice, but we'll be in contact with the people who have expertise in neurological testing, and we will define the best way to assess the aging phenotype.
Mm-hmm. And all the compounds that are listed are planned to be given as the drugs, not like some like food, you know, because I know that like what is there, like experimenting is you know, I don't know, like mushrooms, and sulforaphane it's in broccoli. We're not giving that. We're giving like the pure compounds, right?
Yes, because actually, yeah, so all these natural compounds, they are isolated from, of course, you know, like sulforaphane is isolated from the broccoli, resveratrol. So we don't use resveratrol, but it's another, you know, they're famous in a giant field. The drug, it's contained in the red wine, but actually, so the amount of these compounds that are contained so in the natural products is very low, and you have to eat like, you know, kilograms of broccoli to get the amount of sulforaphane that, you know, really has strong effect. So yeah, we will be so yeah, buying the chemical and adding it to the food. Also pay attention to the to choose only the drugs that are tasteless, because if the drugs will have you know, bad taste for the mice, they wouldn't eat the food. And if for example, so they would be on the other hand attraction too much to the food, we would have the problems with obesity. So we are looking very carefully so on the finding the also compounds that could be safely added to the food without, you know, repulsing mice.
Do you think that this amount of animals is sufficient, or is it just very limited in funding?
Of course, you know, if you look at this, you know, problem from, you know, fundamental, you know, perspective, the answer no, because so there is lots of difference, you know, from the evolution perspective between mice and people. So in the terms of the body weight, in the terms how they, the reproduction strategies, all the things they affect. So the aging. But on the other hand, so unfortunately, at this time, so we don't have ability to make experiments on long-lived species, because, for example, if we take, for example, you know, the bigodog, who live 20 or 30 years, so we will need to perform experiment that will last for 10, for 15 years. And these, you know, long-lived animals, so of course, they will give us information that more relates to the people, but it will take us decades to find a possible combination in this time. So that's why we stick to the small animals. And also, on the other hand, so we can also see that despite all the evolutionary changes between the species, we can see that many features of the aging, so they are evolutionarily conserved. So we can see that, for example, activation of mitophagy can prolong life in flies, in worms, in mice. And so we hope that so we can find how to target these evolutionary conserved mechanism of aging, not because so no doubt there is some species-specific mechanism that contribute to the aging of each specific each specific animal, but we hope that we can find the fundamental these evolutionary conserved mechanisms that we will target, and we hope that our results will be could be translated to the human. But of course, it's just the first step. So if we will see the good results, then we will need additional experiments to confirm the, to make it in other species, to make it in a bigger sampling. And so it's how to say, it's the beginning of the long work. And for the beginning, as a first attempt, I think it's quite good.
I will read out several more questions from the chat. Madiar, he's asking how much do mice cost? What's the biggest budget eater for your experiments? And also, where are you located? Can he come by and see? We know Madiar, in fact, so it's fine. We can vouch for him. So, but sure, you can answer other questions. So what's the biggest budget eater for your experiments?
Budgeted for my experiment. Okay, so I don't remember the figures. That's why I will look in my notebook. Okay, so
the same thing is much is asking the same thing. How much of the budget is routine operations for such experiments, such as work of laboratory assistants? The low qualified work lasting years, is there any cheapening potential by add some automatization in such experiments? What is the most significant financial barrier in scaling this experiment? And yeah, he cannot say it out loud himself.
Okay, so the cost of the mice depends on their age. So the more older mice you buy, so the more they're expensive, obviously, because the facilities that that breed mice, they also spend resources. And so if you, for example, want to buy 1 year mice, it will be $100 per one animal. But so we are going to buy animals that will be 6 months old, and they will spend, they will wait for 6 months in our facility because it will be 2 times cheaper. And actually, so the animals is the, it's the biggest part of our budget. So even with these attempt to save money, as I told you, so buying the animals that half a year and half a year additional waiting for the start of the experiment, so it will take the half of our budget. So it's also depends on the drugs, because for example, so when we were planning the experiment, we wanted to use one drug that is approved to treatment of HIV patients, but it was found in experiments that this drug inhibits the transposon activity, which contributes hugely to the genome instability. But so we found out that so like the whole so the whole our budget will, will be eaten by this drug. So it also depends. So if now we planned our budget in such a way that the half of the budget is taken by the animals, the half is by the medication. But if we, for example, want to make a very small combination, like maybe just 2-3 drugs that will be very expensive, then it will be another ratio. Okay, so what were other questions relating to the financial issues?
Madiar is asking one time, where are you located? Albert Einstein.
Okay, so I'm located in United States, so New York, and so the experiment will be held there, but in general, so our team, is international. So the Roman Litvinov is now working in Russia. Alexei Reshevsky is working in the Ukraine. So Open Longevity is based in the California, and also have people in our community who works in Turkey, in Dubai. So we have, it's, like international effort.
Yeah, but Madiar wants to come and see the whole of the lab and all the mice, the experiment itself. I think Madiar is New York himself, if I'm not mistaken.
Okay, so unfortunately,
excuse me, can I join, right?
Yeah, sure.
Yeah, I just went out of the quiet room when I was not allowed to speak, but now I can. So yeah, you said that unfortunately you do not allow visitors, right?
So it was interrupted.
Yeah, just proceed.
Okay, could you repeat again, please? I'm so sorry.
Yeah, it's okay. So my question was, is it possible to visit your laboratory? Like,
yeah, it's possible to visit, but unfortunately, regarding visiting animals, it's not allowed because so the animal facility controls very good that animals should be free of any pathogens, and so you have to get special clearance to see the animals. So you have to finish short training in order to get the permission to visit the animal facility, but the part of the lab that is not connected with the animals, yeah, you can visit.
excellent. I'm ready to like generally I'm free on Mondays, Fridays, and weekends. So let me know when you're free, when you're available, I would say.
Okay, yeah. So yeah, maybe you can okay. Are you following any of our chats in Telegram?
Yes. I'll text
okay.
you in Telegram.
Okay,
That'll be fine.
Text me then. Yeah, we'll be in contact.
You want to spread the word, Madiar? How to tell the world about our experiments?
Yeah, yes, I would love to. Yeah, I mean,
yeah, that would be amazing. Take pictures,
of course.
What's happening? Okay, great. So right now we have limitation of budget. Well, in terms of what types of compounds we select for the experiment, because several could be theoretically useful but very expensive. All right. So yeah, we would use some help with that, but that's not a must. We can proceed with what we have. Okay, what else? Any other questions, guys? Do you have questions in YouTube, Danila? No, I don't think so. All right.
Okay, so if there is no questions.
Yeah, I just wanted to add, in startup, we are performing the same way as Aubrey is doing right now. We just selected the best that we could afford with the lower budgets that we have than Aubrey does. And then we just proceeded like with the explanation of what types of mechanisms of aging or hormones of aging this thing will affect, and as a result, we can see that they in fact do affect like many, almost all of them, which is great. It is still yes a combination, but as far as I see it, initially it's mostly like budget and affordability, or is it both?
I would say that we have some fundamental, some conceptual differences from the Audrey's design. So one of the things that so we really want to stick with the pharmacological agents because it's the thing that could be easier translated to the people. As you see, so we use in our experiment either the medication that are already available and are already approved for the people, or we used some like pyridoxamine or NAD or spermidine, some compounds that are already naturally occurring in the body, some endogenous compounds, and these are one of the conceptual difference, because you don't need to put additional attempts in order to make these things work to the people. So actually, if we will get their really rigor data showing that we're able to stop aging in mice, then the only thing that we need is to try to do the same in the clinical study, and we don't need to do the toxicological study, because all these things are already done for these drugs when they were initially approved. So because all these compounds are already, lots of, so they're already used in the medicine. So we expect, so we already know very good what side effects patients will have. So it makes the things very easy. And for example, comparing to the genetic therapy for fighting, for contracting telomerization that use Aubrey, or comparing to the treatment with the stem cells. So there is lots of pitfalls because it's totally new things. So you need to do lots of study to prove that it's safe. And also, for example, so with the stem cells, it's a big problem how to scale these, because you need much more cells that you want to inject in the people, and so it makes these potential therapies also very expensive. So using small molecules as therapeutic approach is less expensive. So I think it's maybe the main difference in our strategy.
Okay, so it's more transferable to people then. It'll be easier to run clinical trials. No, makes sense.
Little more practical, I would say.
Yeah, okay. Another question. Do you know, maybe somebody tried this before? We have a combination of vitamin 6, 9 drugs. How many drugs do we have?
Sorry, accumulation of
how many drugs? The combination of how many drugs do we plan to test?
So we plan to use 8 drugs.
Okay, 8 drugs. Were any of them tested in combinations, like in pairs, in like triples?
Good question. So as far as I know, not really.
Well, probably not, yeah. And especially not in the same environment that we are going to perform the experiment. So I really like the approach of Aubrey. He's also including this, like, not only test the bunch of it, like all of them together, but also in pairs. maybe let's, I know we're limited in budget, so we can't afford that right now, but let's maybe calculate the budget for this digital additional feature. So if anyone will want to like fund the improvement of the design, if you do consider this as a potential improvement yourself, we can maybe ask for some additional funding specifically for that. What do you think?
Okay, so the OPRI combination consists of the 4 interventions, and actually, so you can test, in pairs, and it will be like how many 8. So additional experiment for the 8 drugs. If you want to do, if you combine in them pairs, so it will be so many experiments. So for the combination, I think it's. I think it's not approachable at all, but I think what
maybe not all of them, just some of them. Yeah.
No. So I want to say that from my point of view, what could be very interesting, to use this combination for short-term treatment, like for example for 3 months, and then sacrifice animals and do comprehensive analysis what is happening, because for many of the hallmarks we can evaluate using different molecular approaches. We can see how good the activation of autophagy, what's happening with the mitochondrial function, how many senescent cells do we have, and so if we, for example, do this preliminary experiment with a short-term treatment, and after 3 months, for example, we see no big changes with the control group, then we can try to think that, okay, maybe we need to change the combination. And with these, lots of, these short-term treatments, maybe we can really make the combination higher. So, if we are looking for additional funding, from my opinion, I would use it for this. And I would say that, so now we are using like 15K for the whole experiment, but for this short preliminary experiment, I think like 5K will be will be enough. And so if you, for example, can contribute to this sum, we can make these preliminary pretest the combination, try to optimize it before making the long-term expensive experiment.
Mm-hmm. All right, let's put this on paper then. Yeah, because I'm looking at like probably there's always a possibility that the combination won't have an additive effect or a synergetic effect, or it will actually like switch off the effects of, the compounds will switch off the effects of each other. That's also a possibility. So maybe try the combination that doesn't intersect at all in terms of hallmarks of aging. Like Nabilitin and enalapril, they affect the same hallmarks that are being affected by others.
Yes, but okay, so yeah, I work in the biology from 2008, and during this time, so my experience is that 80% of the time we're doing experiments that disproves our hypotheses, and it is this is how it's now in biology. The system, the system is very difficult. The system is very complicated, and we are still, have little information about the organism. And yes, in the majority of the times, we make a hypothesis, we read a lot of literature, we think that, this would work, and we do experiment, and this wouldn't work. And now the problem that without experiments. So, there's a possibility that combination would work, of course, and actually, it's a high probability, but without, going into the experiment, we couldn't know because, unfortunately, so the biological systems are much exceeding our present knowledge about this, and we need to make more and more experiments, and lots of disappointing experiments that are showing how they really work. So maybe my position is that doing experiment is better than not doing. It's better to do and to see that this doesn't work, get to some ideas why it doesn't work, and it will be much more helpful than not doing an experiment, because we afraid that we are not 100% sure in our hypothesis. Because if you 100% sure in the fact, you don't need to do an experiment. This is 100% no. So that's why we're doing experiment, because we don't know.
All right. Thank you so much. That was like a wonderful presentation. In the beginning, you actually were talking about the importance of the research in our field, and usually I like myself skip this part because I've been hearing it for many years. But even in this part, you were able to give some new and very interesting information. So that's kind of cool too. Thank you. And thank you for selecting this combination and actually explaining it. It was great. So yeah, if you have time, let's maybe try to prepare some materials to fundraise if we see that further improvement of the experiment might benefit us. If there's anything to fundraise for, because at that scale, yeah, we can manage, but it's always good to. I think it will be great for our community to see how they can help and how beneficial it will be, if it will be. Yeah, great. Thanks once again. Thank you for everyone for the questions.
Yeah, thanks for attention. Thanks for this opportunity.
Yeah, bye-bye.